Imagine a scenario where foreign cells invade your body, and they don't just, passively float around in there.
Right, they aren't just hitching a ride.
Exactly. They aggressively tunnel into your tissue, they actively seek out your blood vessels, and then they systematically dismantle the cellular lining of those vessels.
Yeah.
And if that wasn't enough, they actually replace your cells with their own.
Yeah, I mean, if you were reading that description on a medical chart, you would absolutely assume you were looking at a deadly pathology.
Easily.
It sounds like, a rapidly metastasizing disease or maybe some kind of parasitic takeover.
But the crazy thing is, for you, and for literally every single person listening to this right now, this exact sequence of events wasn't a disease at all.
Not at all.
It was your very first act of survival. Because this is exactly how the human placenta is built.
It represents the ultimate biological paradox, really.
Yeah.
We are talking about this temporary life-giving ecosystem that fundamentally breaks all the standard rules of immunology and, well, tissue boundaries in general.
Right, because normally if a foreign entity tries to tap into your blood supply...
Oh, your immune system launches a massive scorched earth defense. It's game over.
Yeah.
Yet, when you were just this tiny cluster of cells, you managed to pull off the most complex cellular heist in biology. And your mother's body just allowed it to happen.
Allowed it to happen. It's
Yeah.
wild. Well, welcome to the Deep Dive Lab. Today, our mission is to explore this, what you call the ultimate biological negotiation, right?
Huh?
The human maternal fetal interface.
Exactly, the MFI.
We want to really understand the physical mechanics of how these fetal cells hijack the maternal blood supply without, triggering a life-ending immune attack.
Yeah, and more importantly, what keeps this incredibly aggressive invasion perfectly in check.
Right, because there's this groundbreaking new cellular map that has finally revealed the secret rules of this temporary ecosystem, which we're going to get into.
We are. And, to really appreciate how monumental this new map is, we have to look at what scientists were working with previously.
Set the stage for us.
Yeah.
What did we know before this?
So, for decades, researchers knew the maternal-fetal interface existed, obviously. And they knew that fetal cells, specifically this type called trophoblasts, physically invaded the mother's uterine lining.
Which is called the decidua.
Right, the decidua.
Yeah.
But the actual understanding of how they did this was just incredibly blurry.
Like, how blurry are we talking?
Like looking at a blurry satellite image of a busy city. You know traffic is moving, but you can't see the individual cars or the traffic lights. We knew the mother's spiral arteries, the blood vessels in the uterus, were being remodeled to pump high-velocity blood to the growing fetus. But the step-by-step cellular mechanics, complete black box.
It sounds a bit like knowing a house was, completely gutted and renovated, but you have no idea who the contractors were, or how they bypassed the home's security alarm without setting it off.
That's a perfect way to look at it.
Or like how they managed to knock down load-bearing walls without the entire house just collapsing into rubble.
Yeah, that captures the frustration perfectly. I mean, the maternal-fetal interface is this chaotic, mixed-up battleground of maternal cells and fetal cells just intermingling everywhere.
Right.
Historically, if researchers wanted to sequence the RNA of these cells to see, what genetic instructions they were running, they had to take the tissue and essentially blend it into a cellular soup.
Oh, wow. So just throwing it all in the blender.
Exactly. You would get all the genetic data, sure, but you destroyed all the location data in the process. You knew what ingredients were in the soup, but you had absolutely no idea who was standing next to whom in the actual intact tissue.
So you could see that an invasion was happening, but you couldn't map the front lines?
Right.
You couldn't see the real-time interaction between the invader and the defender.
Mm-hmm.
But that brings us to the breakthrough. Because in an April 2026 study published in the journal Nature, this team of researchers accomplished what honestly previously seemed mathematically and technologically impossible.
It really did.
We're talking about Dr. Susan J. Fisher, Dr. Jingjing Li, Dr. Cheng Wang, and their colleagues from UCSF and Stanford University. They actually built the map.
They built it. And they didn't just map a few cells, by the way.
No, it was a massive scale.
Over 1.1 million single cells. They analyzed them, assigned every single one a maternal or fetal origin, and mapped their exact GPS coordinates in the tissue.
That is insane. How do you even do that? Because you mentioned the blender problem.
Yeah, so the way they pulled this off relies on this advanced technology called stereosec. paired with something called codex multiplex imaging.
Okay, break that down for me. Stereosec.
Right, so instead of blending the tissue into a soup, stereosec is a spatial transcriptomics tool. It basically allows researchers to take intact, microscopic slices of tissue and read the genetic instruction manual of the cells while they were still sitting in their original neighborhoods.
I want to make sure we visualize this correctly for everyone listening. It's like taking a complex fruit tart, right? And instead of throwing it in a blender to see what it's made of,
Right, getting a fruit smoothie.
yeah, a smoothie, you instead have a machine that can scan the intact tart and tell you not only that there are, say, raspberries and kiwis in there, but exactly how they're arranged, and even what chemical processes are happening inside that specific raspberry on the left side of the tart.
That is the exact mechanism at play.
Yeah.
Yes. And then Codex adds another layer to that by painting dozens of different proteins with fluorescent tags on that exact same tissue slice.
Wow.
So by layering these technologies, the researchers literally watched fetal and maternal cells interacting at submicrometer resolution.
That's just wild. And what did they find when they finally put the glasses on and looked at it?
Well, the very first major mechanism they decoded was how fetal cells decide what they are going to grow up to be.
Okay.
The fetal progenitor cells, which are known as villus cytotrophoblasts, or VCTs, face a crucial choice really early on. They can either fuse together to form the outer barrier of the placenta.
Or they go rogue.
Exactly. Or they become the aggressive invaders, which are known as extravillus trophoblasts, or EVTs.
Okay, so VCTs or EVTs. I would assume a transition like that works somewhat like a dimmer switch. Like you have a stem cell that slowly, gradually turns up the dial on its invasion genes while slowly turning down the dial on its progenitor genes until it eventually crosses a threshold. Is that how it works?
You know, many people assumed it was a gradual transition like that, but the spatial map revealed it actually operates as a beastable toggle switch.
A toggle switch, like a hard binary flip.
It is a hard binary flip. When specific transcription factors, these are the proteins that read the DNA and turn genes on or off, when they activate to make the cell an invading EVT, they do two things simultaneously.
Okay, what are they?
First, they activate the entire genetic toolkit needed for tissue invasion. But more importantly, those exact same transcription factors physically bind to the promoter regions of the alternative fate genes and actively repress them.
Wait, so they don't just change jobs, they physically cement over the light switches for the other career paths so they can never be turned back on.
They lock the door and literally destroy the key. It is a mutual exclusion circuit that enforces absolute commitment.
Wow. I guess that makes sense, right?
Yeah.
In a chaotic environment like the maternal-fetal interface, a half-committed cell that gets confused about its identity would be disastrous.
Oh, absolutely.
Yeah.
It has to be all or nothing. So once that toggle switch flips, the cell becomes a fully committed EVT invader, and it begins its march toward the mother's spiral arteries.
Which sets the stage for the actual artery takeover.
Mm-hmm.
And this is the part, this is the mechanism that is just incredibly difficult to wrap your head around from an immunological standpoint.
It really is.
Because the researchers tracked these fetal EVTs as they migrated toward the maternal blood vessels.
Mm-hmm.
And because they had the spatial sequence intact, they caught the exact stages of how the fetal cells displaced the mother's endothelial cells.
The cells lining the inside of her blood vessels, right?
Yeah.
How does that actually happen? Well, it is a literal timeline of destruction and replacement. The map revealed 4 distinct states of the maternal vessel cells during this takeover. It starts with the healthy, canonical, arterial, endothelial cells.
The original homeowners.
The homeowners just doing their job, holding hands tightly with one another to form a solid pipe for blood to flow through. But as the fetal EVTs approach, even before they make physical contact, the maternal cells enter a state the researchers designated R0.
Wait. If they haven't touched yet, how are the fetal cells affecting them? Are they like sending out chemical signals ahead of the invasion front?
Yes. The spatial gradient shows that the chemical proximity alone starts to destabilize the maternal cells. In this R0 state, the maternal cells begin to downregulate their cellular junctions.
Meaning what exactly?
Meaning they literally drop their hands, they lose their physical grip on the vessel wall and on each other. Then, as the fetal cells physically press in, the maternal cells are pushed into the R1 state, where they lose their arterial identity entirely. They just stop running the genetic programs that make them a blood vessel.
So they're essentially forced to forget what they are.
Exactly. And then comes the final stage, R2.
What happens in R2?
Apoptosis. Programmed cell death. They detach completely, initiate a terminal death sequence, and are cleared away by the body's waste management systems.
That is just so the fetal invaders force the maternal cells to unmoor from the wall, forget their identity, and initiate their own death sequence.
Yes. But here is the mechanism that elevates this from a simple attack to a true biological heist.
Right, the identity theft part.
Exactly. While the mother's cells are dying off, the fetal EVT cells undergo a process the researchers call pseudovascularization.
Pseudovascularization.
Yeah. The fetal cells actively begin to run the genetic programs of the maternal endothelial cells. They start expressing the exact same surface proteins.
They literally dress up in the homeowner's clothes.
Yeah.
So the mother's body keeps pumping blood through the vessel without realizing the pipe has been entirely hijacked by foreign tissue. I mean, it is pure biological identity theft.
It is, and the spatial transcriptomics data prove this definitively. The closer the fetal EVTs got to the inside of the vessel wall, the more heavily they express maternal endothelial genes. They systematically adopt a camouflage identity to perfectly mimic the niche they just destroyed.
Which explains why the mother's immune system doesn't attack. Because, as far as her body can tell, those are perfectly normal blood vessel cells doing exactly what they're supposed to do.
Precisely.
Okay, but that camouflage is brilliant, obviously, but it introduces a glaring logical flaw to me.
What's that?
If these fetal EVT cells possess a hardwired genetic toggle switch that turns them into unstoppable invaders,
Yeah.
and if they can remotely force maternal cells to die and completely camouflage themselves from the immune system, why don't they just keep going?
Right.
What stops this from becoming a runaway train that invades the mother's entire uterus or, her surrounding organs?
It's a great question because an aggressive accelerator requires an equally powerful brake pedal. Without it, you would have a fatal, uncontrolled invasion.
So what's the break?
For a long time, the nature of that break was heavily debated. But by looking at the highest resolution map ever created of the invasion front, the researchers spotted a completely novel maternal cell type just hiding in plain sight.
Wow.
They named it the DSC4 decidual stromal cell 4.
Okay, so the DSC4 cells are the defenders. Where exactly are they stationed?
The researchers found them heavily concentrated right at the superficial decidual surface, specifically positioned where the anchoring villi of the placenta press into the mother's tissue.
So right on the front lines.
Exactly. They sit exactly on the front lines, acting as a localized maternal brake on the fetal advance. And they don't do this by building a physical wall, by the way. They use a highly specific chemical mechanism.
A chemical handshake to stop the invasion.
Yeah.
And the specific molecules they use are what make this discovery so broadly impactful, right? Because the DSC4 cells express a receptor called CB1.
The CB1 receptor. Yes, which is a cannabinoid receptor. The maternal DSC4 cells use endocannabinoid signaling. They secrete a molecule called anandamide, which binds to these CB1 receptors to actively suppress the invasiveness of the fetal EVTs.
So the mother's tissue is constantly secreting these specialized molecules to communicate with the fetal cells, essentially sending the instruction, like, you have gone far enough, remodel the vessels exactly here, but proceed no further.
Right, stop here.
Let's scroll back from the microscopic map for a moment, because mapping this perfectly balanced ecosystem has massive implications for real people. Creating a submicrometer map of a healthy, functioning maternal-fetal interface allows scientists to finally understand what happens when the system breaks.
And it does break.
It does. And the researchers figured this out by using GWAS downs, right? Genome-wide association studies.
Taking the spatial map and overlaying GWAS data is just a brilliant analytical leap. GWAS databases contain the genetic information of hundreds of thousands of individuals who have suffered from specific diseases.
And these studies identify the tiny genetic typos or risk variants associated with the condition.
Exactly. But historically, scientists just had a list of typos. They didn't know which specific cells in the body were actually reading those broken instructions.
It's like having a list of spelling errors in a massive library, but having no idea which books actually contain the errors.
Until you build a comprehensive map of every single book, which is what they did. The researchers took the genetic risk factors for severe pregnancy complications and overlaid them onto their new cellular atlas to see where the diseases actually originate.
Let's look at preeclampsia as an example. It's a devastating condition characterized by dangerously high maternal blood pressure. And in maternal medicine, there has been this endless debate over whether preeclampsia is driven by a defective fetal placenta failing to invade properly or by the mother's body aggressively rejecting the placenta.
Right. It's always been this chicken or egg question.
But they mapped the genetic risk to find the culprit. And what they found completely reframes the disease. The genetic risk for preeclampsia isn't isolated to one side of the equation.
No, it is fundamentally a failure of the cellular handshake we just discussed. The researchers found that the genetic risk factors originating from the fetus mapped specifically to those invading EVTs.
Okay.
But the genetic risk factors originating from the mother mapped to a specific maternal cell type called DSC3, as well as her arterial endothelium. Preeclampsia is a coordinated breakdown.
Wow.
Yeah. The fetal cells possess flawed genetic instructions for how to invade, and the maternal cells possess flawed instructions for how to receive them. Both partners in the negotiation fail simultaneously.
That dual failure mechanism changes how we have to think about treating it. You can't just target the mother's blood pressure. You have to look at the interface itself.
Exactly.
And they applied this same mapping technique to other devastating outcomes like sporadic miscarriage and spontaneous preterm birth.
They did. And when they overlaid the maternal genetic risk for both miscarriage and preterm birth, the data didn't scatter randomly across the tissue. It zeroed in on a single, highly specific maternal cell population.
Which one?
The risk mapped to endometrial epithelial cells marked by the genes POU5F1 and LGR5.
POU5F1 and LGR5. Finding a single cellular origin point for both conditions is a massive shift, because those specific epithelial cells are present very early in gestation, right? Long before a miscarriage or a preterm birth actually happens in the clinic.
Exactly. Because these conditions share this fundamental vulnerability at the cellular level, the researchers actually proposed a new classification, endometrium spectrum disorders.
Oh, interesting.
Yeah, the actual mechanism of a miscarriage or a preterm birth appears to be baked into the very early structural foundation of the uterine lining. The architecture is flawed from the moment the blueprint is drawn.
Which means future treatments might focus on identifying and correcting these structural deficits before the fetal invasion even begins.
That's the hope.
This research obviously reshapes our understanding of disease, but I want to return to the maternal brake mechanism for a second, the DSC4 cell.
Okay, yeah.
Because understanding the mechanical biology of that specific cell solves a major public health mystery. The mother's natural chemical brake pedal relies on CB1 cannabinoid receptors. And that biological reality perfectly explains why cannabis use during pregnancy is linked to adverse outcomes.
It does. If we look purely at the cellular mechanics, the active ingredient in cannabis, THC, is a potent CB1 agonist.
Meaning what?
That means it is molecularly shaped to fit perfectly into those exact same cannabinoid receptors on the DSC4 cell.
So it operates through competitive binding. The mother's body is trying to send this delicate, highly calibrated chemical signal using anandamide to tell the fetal cells to stop invading. But if a pregnant person consumes cannabis, THC floods the front lines. It basically jams itself into all those CB1 locks.
It completely overrides the biological instructions. By artificially flooding those receptors with an external molecule, you dysregulate the delicate endocannabinoid signaling at the invasion front. You essentially jam the maternal brake pedal.
Wow.
And this mechanistic discovery seamlessly aligns with what epidemiologists have observed for years. Prenatal cannabis exposure is strongly associated with spontaneous preterm birth, low birth weight, and severe placental dysfunction.
So we've moved past epidemiological correlation here. We aren't just observing that two things happen at the same time. We have the exact cellular mechanism. The THC overrides the molecular stop sign.
Right.
It's a perfect example of how mapping the normal, healthy function of a system instantly illuminates why external chemicals can throw it so dangerously off balance.
It reinforces just how temporary and incredibly delicate this ecosystem is. Every single cell relies on a perfectly timed sequence of chemical signals just to survive.
It's amazing.
Yeah.
Well, as we distill this deep dive into the maternal-fetal interface, I think there are three fundamental takeaways about how, you survived your very first days.
Let's hear them.
First, fetal cells are master mimics. They utilize a hardwired, biasable genetic toggle switch to commit entirely to tissue invasion. Then they orchestrate literal biological identity theft, forcing the mother's blood vessels to unmoor and die off, while simultaneously camouflaging themselves with maternal genes to steal the blood supply undetected.
That's right. And second, the mother's body actively manages this aggressive invasion. It is not a passive environment. It deploys localized chemical brakes, specifically the newly discovered DSC4 cells, which use delicate endocannabinoid signaling to draw a definitive line in the tissue and halt the fetal advance.
And third, by mapping this cellular dance at submicrometer resolution, science has finally pinpointed the physical origin of major pregnancy complications. Preeclampsia is a two-way breakdown of the cellular handshake, while preterm birth and miscarriage are rooted in early structural flaws within specific endometrial cells.
Seeing the mechanics of this system mapped out so perfectly really leaves us with a profound concept to consider. Think about it. We have just spent the last several minutes describing a highly orchestrated process where foreign cells safely invade adjacent tissue. They aggressively remodel blood vessels to secure their own nutrient supply, and they completely alter the local immune system so they aren't destroyed on sight.
Wait, you're describing the exact same mechanistic playbook used by cancer. Tumors invade tissue, trigger the growth of new blood vessels, and actively suppress the immune system to survive.
Wait, the parallels are undeniable. But the difference is that the placenta executes this invasion perfectly, safely, and then crucially, it knows exactly how to stop.
It turns the invasion off.
Right, it turns it off. The tumors do not. So what if the mechanisms mapped out in this study, the genetic toggle switches, the targeted apoptosis of blood vessels, the localized chemical breaks, what if they are the ultimate biological blueprint?
Wow.
If we can fully decode how the placenta safely orchestrates and contains an invasion, could those same chemical instructions teach us how to permanently shut down the deadly, unchecked invasions of cancer?
The idea that the very first act of human survival might hold the molecular keys to curing our most complex and devastating disease is a staggering thought. It fundamentally changes how you look at the beginning of life.
It really does.
Well, thank you for joining us in the Deep Dive Lab today. If you enjoyed unpacking the mechanics of this biological mystery with us, be sure to follow and subscribe for more fascinating science stories unfolding in real time. We'll see you on the next Deep Dive.